Hi, everyone. Thank you for joining today's webinar. I'm Le Ying. Today, I'm excited to walk you through how to process image data to achieve single-cell resolution gene expression on Stereo-seq. We will cover detailed steps on how to use the high-resolution imaging to get single-cell results spatial matrix. In pathology, high-resolution images like those from H&E staining or fluorescence images are important. They help pathologists detect subtle abnormalities, assess tissue heterogeneity, and confirm disease states. These staining images capture the overall tissue architecture, the spatial arrangement of cells, and the extracellular components. This level of detail is essential for identifying tissue regions and substructures. And also, these high-resolution images allow us to observe the morphology of individual cells. When we combine these images with spatially resolved transcriptomics data, we can analyze gene expression at the cellular level. This integration offers a deep insight into tissue function and disease progression. With Stereo-seq, the images and the spatial gene expression matrix come from the same sample slice. The staining images give us detailed histological features, while the spatial matrix reveals the gene expression activity across the tissue. Image data plays a pivotal role in delineating tissue boundaries. For example, as shown on the slides, the upper row demonstrates how we identify tissue boundaries from the image, and the lower row shows the corresponding spatial matrix result. The image-based recognition is more precise in defining these boundaries. We can see here and also this eyeball data. Additionally, the detailed cell shape and size information from the images is used to guide the isolation of individual cells. This is the first critical step in achieving single-cell resolution in our spatial transcriptomic analysis. In today's session, we will cover the following topics. A walkthrough of how we process microscope images to generate a spatial matrix at single-cell resolution. Explain the tools involved and share some insights into overcoming current challenges. Let's move to the first section. We begin with image acquisition. First, high-quality imaging is the foundation of our analysis. Clear images reveal tissue structures, cell positions, and spatial markers that guide the integration of gene expression data with tissue morphology. In our Stereo-seq experiment protocol, tissue slices are mounted on a Stereo-seq chip slide and stained using the nuclear or H&E stains before imaging. Note that the Stereo-seq chip is opaque, may make the image slightly dimmer than those captured on glass. Because the image quality is highly dependent on the microscope and imaging conditions, some requirements are necessary to achieve optimal results. For reliable spatial analysis, it is vital that all targets, here we need the tissue, cells, and also the alignment markers, our track lines, are clearly visible. Tissue staining images provide critical information about the tissue structures and the cell locations. The image must show complete tissue morphology with distinct boundaries. If the single-cell resolution spatial data analysis is expected for downstream analysis, the images must also clearly show each individual cells. The Stereo-seq chip surface contains the track lines, which are the perpendicular lines that are arranged at periodic intervals. These track lines are visible in Stereo-seq data, appear as narrow lines on the spatial heat map like this. This is the zoom-in view here, and also in the image where the tissue uncovered. Here is an example for the HE and an example for the ssDNA. We can see that in the H&E image, the track lines show in the white, and in the ssDNA or DIP similarly, they show in the black. The track lines are used for highly precision alignment of the microscope images with the spatial feature expression matrix. To achieve this, we require that the image must contain a sufficient number of track lines forming a grid pattern like this, the grid pattern that can be used as an alignment reference template. Meeting this criteria requires specific microscope and imaging procedure settings. To ensure the compatibility with Stereo-seq analysis, the imaging systems must meet specific requirements. Here shows some requirements for your microscope. The fluorescence and color imaging have slightly differences, but the core principles remains the same. There are a lot of requirements here, but I will further explain some of those together with the imaging procedure requirements. For Stereo-seq image acquisition, we will require a 10 times objective magnification with a pixel size of 0.3 to 0.75 micrometers. This ensures a balance between the clarity, the acquisition time, and the file size. Since the cell nuclei are the minimum size of the cell nuclei can be around like 5 micrometers and track length about 1.5. So the high resolution is necessary to distinguish these features. And next is the focus. The tissue sizes are slightly elevated on the Stereo-seq chip. You may need to create multiple focus model points to ensure that both the tissue cover region and the uncovered region are in sharp focus because we need both the cells clear and also the track lines clear. Also, we need the movement of the microscope is stable, and also we have good staging. This is because the microscope imaging involves capturing multiple small regions and staging them into a complete image like this. A stable movement and the precise staging prevent the distortions. Also the discontinuities of the tissue like this. And misalignment between the cells and spatial expression matrix because the track lines can not be like a straight line. Also, we require a proper contrast and exposure settings, which is crucial to highlight the tissue details. The overexposure or underexposure can obscure cells and track lines, which will affect the alignment accuracy and also the segmentation. Then for the track lines, the image must capture enough track line intersections to form a grid pattern that allows for measuring the track line intervals in both the X and Y direction. Just like the green square shows here, the 5 track line intersections could form an acceptable grid. Like this. The 5 track line intersections form this grid. The other 2 examples are not acceptable because the intervals cannot be measured like this. They miss at least one of the lines to form this grid. To ensure the correct orientation and avoid unintentional mirroring in the final image restoration, it's recommended to make sure that your export image has the same orientation as the tissue's actual orientation on the chip when the chip label is positioned on the right side. By following all of these imaging guidelines, we are like 60% done with the cell beams, almost 70. We create high quality images that serve as a reliable foundation for the registration and segmentation in the next steps. Now we have got our images. So then we will move to the actual image processing steps. Stereo-seq image processing consists of 4 main steps: the image QC, the image registration, and the tissue segmentation and cell segmentation. We provide automatic tools to streamline these steps along with minute tools. The first step is image quality control. Image QC is primarily determined whether the track lines can be identified from the image, as they are crucial for high resolution automatic registration. If an image fails QC, we will recommend to retake it if possible. Otherwise, you may need to do the registration rely on tissue morphology, which can be quite time-consuming and cannot ensure the accuracy. After QC, we can move to the registration step. This is almost most important step in the entire image processing workflow, as it adjusts the image orientation and align it with the spatial matrix with high precision. Also, it determines whether the subsequent matrix segmentation based on the image recognition is accurate. Here, I will explain each step in detail. The first step of image registration is to level the image by adjusting the rotation angle to align the track line horizontally. Then the image is scaled according to trackline based rules derived from the Stereo-seq chip number. Here we have fixed the small angle rotation issue here. After that, we move to the next step, which is the flip. Flipping is a mandatory step in the automatic workflow. This is because the chip mask file, which containing the X and Y coordinates for each transcript, has a different coordinate origin than the image. Here, the left bottom corner is the origin of the chip mask, and the left top corner is the origin of the image visualization coordinates. Yeah, so they have the flip issue here, so we do the flipping for our image. However, the track lines alone cannot verify whether the flip was applied correctly during the QC. So probably we can have some pre-check to this. To avoid flipping errors, we would recommend you to strictly follow the imaging guidelines. Ensure the exported image maintains the same orientation, including the rotation and the flipping as the actual tissue placement on the chip when the chip label is on the right side. By following this, you can avoid flipping issue in the final registration result. After flip, we will move to the step 3 and 4. From here, the spatial matrix is involved. Step 3 is quite straightforward, that the image is roughly matched to the matrix by aligning the center of mass. This step recalibrates the image's coordinate origin, ensure consistent spatial referencing. And then in step 4, the image is rotated in 90-degree increments, like 0, 90, 180, and 270. For each rotation, the overlap between the image and the spatial matrix is assessed, and the angle with the highest overlap rate is selected as the final orientation. We have done the rough center matching. The step 5 performs precise cell level alignment, which is achieved using the pan movements based on the track lines. After that, in step 6, the image is cropped and padded to make the image and matrix share the same shape and size, and also the coordinate system. So now we have got our registered image. Then the next steps are tissue segmentation and cell segmentation. Tissue segmentation identifies tissue boundaries, while the cell segmentation focuses on detecting each individual cells. Both processes generate the mask files, which are the binary files that distinguish foreground targets from the background. Note that for the tissue segmentation, we also do another step, that is we remove the cells that are not covered by the tissue mask. So we only left the cells within the tissue region. Now we have all done for the image processing steps. Then we will move to generate the cell bin. So what is cell bin and why we care about it? Cell bin is a type of spatial unit. A cell bin, unlike a square bin, like bin 1 or bin 20, bin 5, bin 50, the cell bin is defined based on the contours of cell morphology rather than a fixed grid. by merging adjacent spatial beams and integrating feature expression signals. Cell beams forms spatial units that enable precise single cell resolved spatial transcriptomics analysis. For example, cell type annotation, tissue microenvironment analysis, cell-cell interaction studies, and so on. Get cell beam matrix is similar to make a cookie. If you cannot understand this process, you can just imagine that we are making the cookies. Your cell mask is like the cookie cutter, and the dough and our spatial matrix is like the flattened dough that we use the cookie cutter to extract a specific shape from the dough. That is how we get the cell beam matrix. Since the ssDNA, DIP, and H&E images provide only, I mean, not only, but just we do the cell nuclear segmentation, we only can identify the nuclear boundaries information from these images. An additional post-processing refinement step is applied during the cell beam generation. In our Stereo-seq workflow, the cell region is expanded by approximately 10 micrometers to capture relevant transcriptomic signals while preserving precise cell locations. As shown here, the red circles are the nuclei mask, and the white lines are the expanded cell mask. Okay, let's conclude the process of generating the cell beam. But what if adjustments are needed, and what tools are available in Stereo-seq data analysis software? Let's move to the next section, tools and tips. In this section, I will introduce key tools in SW and Stereo-map, along with commonly used cell segmentation algorithms. Additionally, we will discuss image abnormalities, what issues can be corrected using manual tools, and what needs to be addressed during imaging to ensure accurate results. Stereo-seq image processing is powered by two key tools, the SW and the Stereo-map. SW provides an automatic pipelines handling the registration, segmentation, and target matrix extraction for QC pass images. It aligns the image with the spatial matrix using checklines and generates both the tissue matrix and cell beam matrix, the tissue matrix and cell beam matrix for the downstream analysis. Meanwhile, Stereo-map is a desktop application, provides manual adjustment tools. It offers an interactive interface for like fine-tuning image orientation, scale, and location. That is what we need to do for the registration. And also with lasso and drawing tools, you can manually create tissue and cell masks. If the default tools don't meet your needs, you can also import and replace masks generated by the other tools or algorithms. Additionally, the Visual Explorer allows for region of interest extraction for more customized analysis.
Now I'm going to show you how to use Stereo-map image processing module to do the image registration and segmentation. Here I've already imported an image into the Stereo-map. Once your image is loaded, simply click Next to proceed to the next step, our registration. In this step, you will find several image adjustment tools that allows you to adjust brightness, contrast of the image to enhance the tissue morphology and the visibility of the track lines. Like this. You can see the track lines here. Yeah, and also the tools like rotation. You can rotate the image. Flip, and also the scale in the horizontal or vertical directions. And also the pen move. You can move upper right, you can also change the step size. For example, let's see, 40. Yeah. For the QC past images, you can use chip track line representation, which is this yellow crosses to check if the alignment is correct. The reason we provide this crosses is that during the registration, the heat map is overlaid on the image, so you can't see both the track line and the matrix track line and the image track line at the same time. So instead, we this yellow crosses could act as a representation of the matrix track lines that can be used to help you match them with the image. A good registration means that the intersections of these track lines overlap accurately. To be sure that everything is well aligned, I recommend taking at least 2 zoom-in views diagonally across the tissue. Like here is the top right view around the tissue. We can see the track lines are overlapped. And also we go to the left bottom view. We can see here the track line is also overlapped, so we can see the registration is good. However, if you're working with the QC failed images, the things can be a bit trickier because the track lines cannot be clearly visible or usable. So in this case, we need to adjust the brightness and the contrast, or use the normalization for quick adjustment to make the morphology of the tissue from the image clearly visible. Here we can see the feature of the matrix, and also the morphology from the tissue can be clearly seen. So the thing we need to do is to adjust the image to make the feature aligned. Once you have done with the registration, click next to the tissue segmentation step. Now moving on to the tissue segmentation step. Here you will see a purple colored region. This represents the tissue mask. You can use lasso, brush, and also the eraser tools to edit this mask and refine the segmentation. The lasso tool is great for selecting or deselecting large areas quickly. And also the deselect. Yeah. The brush and eraser tools, you can also change the size of it. They are better for precise adjustment. For example, for the tissue edges, you can use the brush to draw, and also if I want to include this cell, you can use the pen. One important thing to be noted is that the tissue region you define here will directly impact the next step. For example, if I exclude, just use this, use the eraser here. If I exclude this region from the tissue mask, the cells under this region will not count it. Let's see. If I think this is my tissue mask and click next, you can see this region will not be counted. This part will be removed. Okay, now we are at the cell segmentation step. Here we can see the cells are shown in the red, and also they are not the field mask. Instead, we use the use a outline to show each individual cells for the better realization. Similar to the tissue segmentation, cell segmentation also have the lasso, brush, and eraser tools. I already use the deselect lasso to remove a large region a bunch of the cells, and use the brush to merge some of the cells. For example, I think this is one cell here, so I just use the brush to merge them. And if I think that should be the two cells, I can use the brush to separate them. Yeah, as shown here. Or if I don't like the current mask, I don't want to make fix on it, I can try to import a new one. For example, here I click the mask, let's create a new one, or I can click the plus or replace to upload a new TIFF format mask here. See, I have uploaded a new one, and you can see that this is a completely new mask here. Keep in mind that even we import a new cell mask, the region we removed from the tissue segmentation will still excluded like this. If we don't want this, we can go back to the tissue segmentation step again, using the brush to fill it, or we can also use the lasso to do a quick fill. Then we can click next to see what happened to our cell segmentation result. We can see that this region has been included while still use the new imported mask. Okay, now we're done with all the image processing steps. Then we will go to the final step, which will export the manually processed image to a tar file. This file can be then passed to the SCW count or the realign pipeline to do the tissue segmentation or the cell segmentation. Here is the path of the exported image.
So by combining the StereoMap's annotation automation with StereoMap's flexibility, you can achieve accurate and precise image processing for your Stereo-seq data. Okay. As I just mentioned, that StereoMap allows you to import segmentation mask files from other tools. Next, I will discuss 3 types of segmentation tools commonly used in the spatial analysis. They are like the manual tools, the drawing tools, and the semi-automatic segmentation tools, and the fully automatic, usually deep learning-based methods. The manual segmentation tools such as the lasso, paintbrush, eraser, as I showed in the map, offer complete control over the segmentation process. This type of tools are ideal for small data sets or for fine-tuning errors produced by automatic segmentation methods. However, because these tools require cell-by-cell adjustment, they are very time-consuming and not feasible for larger data sets. Thus, while they yield high precise results, manual segmentation is best reserved for refining results or addressing specific edge cases. The semi-automatic segmentation tools, such as QPath, offer a balance between the automation and user control. These tools allow you to adjust parameters for tissues with variable cell density, size, and also shapes to optimize the overall segmentation outcomes. Finally, the deep learning driven tools such as deep cell, the cell bin, which is the built-in algorithm in SW, and also the cell pose. They offer the fast, scalable segmentation for large data sets and complex cell morphologies. While they enable high throughput image processing and provide significant adaptability, they generally require training and model tuning before using. Therefore, these tools are best suited for the bulk processing tasks. though they can be more computationally intensive compared to other two methods. As a conclusion, to get an optimized result, probably you will need to combine these approaches using deep learning for bulk segmentation first, and then use semi-automatic tools for refinement, and finally use the manual tools for detailed collections. Definitely the easiest method is the fully automated tools. But each algorithm here has its own unique characteristics. So next, let's see a few different examples. Here are some scenarios arranged from easiest to hardest to show how each algorithm performs. The first row is an ideal condition where the cells are clear and sparse. All three algorithms perform well and accurately identify the cells. The second row is a low contrast condition. Under the low contrast, performance varies. We can see that the cell pose misses many of the cells. And cell bin V3, which is our algorithm, fails to identify any actual cells. The third row is a dense cell region, where the cell pose again loses many cells, whereas the deep cell and cell bin perform similarly and successfully identify most cell regions. Now let's see three more challenging scenarios. The blurry images. In images with significant blur, segmentation becomes quite hard. Cell pose tend to be conservative, ensuring that only the regions with clear evidence of cells are marked, whereas the deep cell and cell bin are more inclusive, often labeling ambiguous regions as cells. Actually, cell bin is like in between, not like deep cell have more like background regions. Next is the non-cell substance with stronger contrast. When the non-cellular material appears with higher contrast than the cells, all three algorithms missingly include these regions as the cell. And finally, the last example is the irregular shaped cells. In areas where cells have irregular shapes, cell pose tends to delineate round cellular boundaries. Meanwhile, the deep cell markers any potential regions as a cell, resulting in a broader but less specific segmentation. All above examples illustrate that algorithm performance can vary significantly with different imaging conditions, and no single algorithm is the perfect. Let's now turn our attention to image abnormality. While many tools can fine-tune segmentation in some cases, they cannot fully compensate for imaging issues that obscure or distort critical cellular details. For example, the blur, overexposure, and uneven brightness can cause the loss of essential cellular details, making it nearly impossible for segmentation algorithms to distinguish individual cells. These problems are best to resolve at the imaging stage. Some abnormalities, however, can be addressed through post-processing or advanced algorithms. For example, the background impurities. It can be corrected using the tissue segmentation. Just remove this background. While non-cellular structures, such as the fibers or clumps, and also the cells with unusual morphologies, can be handled by developing and training deep learning models to differentiate and exclude non-cellular structures while recognizing the unique shaped cells. There is also a type of image that recently showed that the region outside the chip has a stronger contrast than the tissue. The white region, which should be which will be identified as the like foreground because they have the similar color as the tissue. So for this condition, you could probably like cut the edge and leave the tissue side. the tissue regions. The last part will be some challenges and emerging solutions to the image processing. Stereo-seq offers three core products: Stereo-seq FF, Stereo-seq Omni, and also the Stereo-seq. And they support overall they support three staining types: SSDN or DIP, H&E, and multiplied immunofluorescence. Each product supports different staining types, as shown in this table here. To be noted that the IF image do not display track lines, so the automatic alignment using track lines is not supported. Instead, we assume that all the IF images should be perfectly aligned with the corresponding DIP image. So the DIP could serve as the alignment bridge between IF and the spatial matrix. In the Stereo-seq DIP and IMIF product, there are no restrictions on F signals, which means that we cannot use the F to do the cell segmentation. But here we use the DIP segmentation that within the region of high F intensity as the F cell result. For Omni products, though automatic HE alignment is supported, the cell bin generation is not enabled by default. This is because the FFP samples show high variability in preservation, making the image hard to be used for the cell segmentation. As an alternative, you can use third-party segmentation results and import them back into the workflow for further analysis. Despite advancement in Stereo-seq image processing, there are still several challenges remains. For the alignment challenges, the symmetric tissue shapes may introduce orientation ambiguity, making it difficult to determine the correct orientation angles. Here, like this two example here, and also the tissue pattern differences between the image and the spatial matrix. The tissue outline in the image may not perfectly match the spatial matrix due to the variations in the gene expression patterns, such as this place. In the spatial matrix, the expression is quite low and close to the background. And in the image, we can see the tissue structure here. So this difference can affect the alignment of the mass centers between the image and the matrix. For example, the center of mass of the image is probably at here, and the matrix probably here. So when we do the matching for the center of mass, they can be quite far away. The alternative way, we have an alternative registration method to address these two challenges. We recently released a feature point registration function in Stereo-seq. This method relies solely on the track lines and can be applied before generating the spatial matrix. The only two requirements that need to be strictly followed are the image should pass QC, which ensure the track lines are visible, and the image orientation is same as the tissue orientation on the chip when the chip label is on the right. So you can just when you do the registration, you can only select the left bottom points here that will be used for calculate the alignment parameters. And also we have some segmentation challenges. For the segmentation, due to the extensive diversity in macroscopic images, also different cell shapes, different microscopic conditions, staining variations, all could complicate the segmentation process. And there is no one-fits-all algorithms. Those can be our segmentation challenges. To enhance the segmentation accuracy and address current limitations, integrating multi-omics and multimodal approaches could be a potential solution. Currently, we are using the nuclear staining and the expansion to identify cell locations, actually the nuclear locations, and expand to get a rough estimation of the cell regions. But what if we can add the cell boundary staining? It could help define more accurate cell size and shape, and also integrate the HE images, which could add morphology details and help distinguish for different cell types. These are the traditional segmentation methods. Beyond that, the AI-driven models can further improve image processing, probably like developing versatile models that generalize across different staining types and tissue conditions, and also utilizing self-learning models that continuously refine segmentation accuracy based on feedback and new data sets. Okay, here are the talk we have today.